Much of the research in this IRP focuses on genome to phenome mapping. This means understanding how the information in the plant genome is translated to internal phenotypes, such as molecular profiles, and external phenotypes like biomass production under different environmental conditions. In this context, we look at the plant microbiome as an associated plant phenotype. Major biomolecules of a cell include RNA, proteins, and metabolites, and these change depending on their biotic and abiotic interactions. Molecular phenotyping, consisting of transcriptomics, proteomics, and metabolomics, helps us to characterize these biomolecules. These data provide the information needed to correlate microscopic molecular changes to macroscopic observations and important parameters related to plant health and vigor. We grow plants in the EmCell Phytotron, which is a state-of-the-art plant growth facility. This allows us to grow plants under tightly controlled environmental conditions. For example, we can impose drought and we can simulate future climate scenarios. Much of these analyses are being conducted on various capability platforms across EmCell. Also located here at EmCell are world-class mass spectrometry and NMR capabilities. Our expert scientific workforce, who specialize in above and below ground plant studies, use this collection of instrumentation to perform cutting-edge plant and soil microbiome research. We are linking genotypic information to internal and external phenotypes using both model plants and bioenergy crops such as sorghum. By doing this, we are trying to narrow in on the genome to phenome gap. Our ultimate goal is to get a predictive understanding of the plant microbiome soil system in transition. One such example is the stable isotope tracer studies we use to follow carbon as it gets fixed, incorporated into different biomolecules, and partitioned through the plant itself and leached out into the microbiome and the surrounding soils. We are basically observing carbon on the move. One important area for future capability development is to significantly increase the throughput for plant molecular phenotyping. For example, to speed up analysis times at the mass specs and develop robotic systems for high throughput sampling and sample processing. Using the Phytotron, we are also developing non-destructive root and leaf phenotyping, as well as stable isotope probing of carbon and nitrogen. Collectively, all of this research we conduct here is important for understanding of these biological processes in plant microbe soil systems. This helps us to create models to understand and predict changing environmental conditions and prepare for a sustainable future.